this is going to be the last session in terms of the series of sessions on ear, on hearing and balance. So with this we are going to include the physiology of balance and equilibrium. Uh, this session would talk mainly about, we are going to start with the structure, and we have already done the structure of the semicircular canal, but we would have to revisit a little bit of the structure, normal stimulus, transduction at the receptor level, and function of the semicircular canals. Then we are going to talk about the otolith organs, which are basically the utricle and saccule. So we will talk about the normal stimulus, transduction at the receptor level, or the otolith organs like utricle and saccule. Then we are going to talk about the central connections, the vestibular nuclei, and the pathway of the vestibular system, and where are these connections going and what they are doing. And the last piece is a little bit complicated because we would talk about the vestibulo-ocular reflex and the pathway of the vestibulo-ocular reflex. And then we would also try to find out the lesions in the brainstem using the calorie test, and the basis of it is the vestibulo-ocular reflex. So that is something which is high yield, usually asked in board exams a lot because it is complicated and you need to understand it. And finally, we are going to do a little bit of clinical correlations with the vertigo. Already you have a clinical lecture, so you have been given a high dose clinical aspects of vertigo, but we would do a little bit of so I think it would start to make more sense now because you already have the anatomy and you already also have some clinical background on certain diseases. Then you can actually put them together, so it would start to put some sense why we are learning this physiology. So the vestibular system is basically one of the three sensory systems which is responsible for your spatial orientation and posture. So it's not the only system which is responsible for your spatial orientation. You have two other systems, sensory systems. One is the vision, and the other is your somatosensory system, which is bringing the information from your joints, from your tendons, that where your body is located or what is the orientation of your body. So the integration of all these three systems, your vestibular system, which is basically looking at the position of your head, okay, your eyes, and your somatosensory system, which is basically bringing the proprioceptive impulses, is basically going to tell you where your body is there in terms of the spatial orientation and posture. The three stabilizing systems overlap sufficiently to compensate for each other's deficiency. Now, in terms of the vestibular system, it is working on two issues. One is the gravitational pull, and the other is your rotational movements, which stimuli, or these are the stimuli which are going to cause the stimulus in your vestibular apparatus, including utricle, saccule. They are mainly affected, utricle and saccule are going to be affected by gravitational pull or linear acceleration. That is how the macula in the utricle and saccule will be affected. For rotational movement of your head, it is the semicircular canals which are going to be affected. So this is just a diagram showing you that all these three sensory inputs, visual, vestibular, and somatosensory, are going to come together to give you a spatial orientation, and they overlap with each other so that you can compensate for deficiencies of one system with the other. The sensory systems which are located, or sensory receptors which are located in the semicircular canal, we said that the part of the semicircular duct which is opening into the utricle, before it opens, it dilates. It forms a ampulla. Within that ampulla, you have the sensory organ of the semicircular ducts or canals, and that is called cristae or crista ampullaris. So you can call it crista ampullaris because it is in the ampulla. And the sensory organ in the utricle and saccule is called macula. So macula is the sensory organ in the utricle and saccule, and crista is the sensory organ, or crista ampullaris is the sensory organ which is present in the semicircular ducts or canals. Now, if you have damage to these sensory organs, either in the semicircular canal or in the saccule and utricle, this will cause vertigo and nausea. So any head movement in if these areas are damaged can result in causing vertigo and nausea. Vertigo is spinning of your head. So the vestibular apparatus, all this vestibular apparatus is present inside the petrous temporal bone, which is the bony labyrinth, and inside that is the membranous labyrinth. So we were talking about saccule, utricle and semicircular ducts. These are membranous labyrinth. Outside them there is a bony labyrinth made up of vestibule and semicircular canals. Between the bony labyrinth and the membranous labyrinth, you have this is the bony labyrinth, and inside the bony labyrinth is the membranous labyrinth. Between the bony labyrinth, which is this bone, and membranous labyrinth, you have perilymph, and inside the membranous labyrinth, which is making the cochlear duct and then the saccule and then the utricle, there is endolymph, which is potassium rich in concentration. Okay. Now let's go back a little bit. Going to go back. Let's define equilibrium. Somebody was asking, there are two states of equilibrium. So equilibrium is a state of balance that allows us to position our body in a three-dimensional space under normal gravitational conditions. So if they are normal gravitational, we are not in the space. We are basically in normal gravitational conditions, and the state of equilibrium, makes our body to balance ourselves. Adequately in a three-dimensional. We are not falling on this side or falling on the other side. We are balancing ourselves under normal, gravitational conditions. That is a state of equilibrium. How we balance it? We have two, components of equilibrium. One is that when we are moving, you are moving, you're running, or you're turning, so you can maintain your balance. This is called dynamic equilibrium. Okay, that is the function of the semicircular ducts. When you are moving, your head has to move also. When you are moving, you're running, your head is moving up and down. When you are turning, your head is moving, turning and round. So when you are moving or rotating, you still are able to maintain your equilibrium. You can fix your eyes to a target. You can, even if when you are running, you can fix your eyes to a target. It doesn't, your image, because your eyes are moving up and down, it should, the image would fall on the retina, should be up and down, but somehow you are able to fix on the target. The image is there and you are able to maintain your balance while you are running, while you are rotating. So this is basically dynamic equilibrium. Now you are stationary, and your head moves forward or backward or move this way or that way. You still are able to fix on the target. You still are able, your head moves this way, you don't fall on this side. Your head moves on this way, you don't fall on that. Your head moves this way, you don't fall. Even you are stationary. This is static equilibrium, or the gravitational pull, like you are in an elevator. You are stationary. You're going down. With the gravity, your head is going down, or you're going up. This is against the gravity, your head is moving up, but you are stationary. This is static equilibrium. This is the function of utricle and saccule. When you are not moving and your head position change, you maintain your balance, you fix yourself to the target, with whatever you are seeing. That is the function of utricle and saccule. When you are moving and your head is changing, your position of your head is changing, or you are rotating, you are in a dynamic state. That is called the dynamic equilibrium. That is the function of the semicircular canals. Everybody clear about the difference between static equilibrium and dynamic equilibrium? Okay. So rotational movements are detected by hair cells found in the ampulla linked to the semicircular canal. Gravitational changes are detected by otoliths, organs found within the maculae present in the utricle and saccule, which are when you are static, you are not moving. Okay. So first we are going to talk about the three semicircular canals, and we can see here you have, this is the cochlear duct, which is opening into the saccule, and the saccule is opening into the utricle, and within the utricle you can see the three semicircular canals which are opening. The usually semicircular canal word we have used, both for the bony and but usually the accurate nomenclature is that the bony labyrinth is semicircular canal, and the membranous labyrinth, which we are going to talk today more, is basically the semicircular ducts. Okay, accurately you should call semicircular ducts, but people keep on using canal and ducts alternatively. Canals are the bony ones, and ducts which are inside them are basically the semicircular ducts. Now there are three major semicircular canals or ducts, or whatever you want to call them, and we are going to see, the first thing which you need to see is their position. So this diagram is very important because you need to understand the position of these three semicircular canals or ducts. So the anterior one, or the superior semicircular canal, if you see the petrous temporal bone, it is like this. Okay, your sagittal plane is, if I make a sagittal plane, this is going to be like this. This is my sagittal plane. So the anterior semicircular canal to the sagittal plane, if this is the sagittal plane, it is making an angle of 41 degrees with the sagittal plane. This is the sagittal plane. The anterior semicircular canal is not straight. It's like this. So it's making an angle of 41 degrees with your sagittal plane. If you look at the posterior semicircular canal, it is like this. This is your sagittal plane. This is the posterior semicircular canal, which is making an angle of 56 degrees with the sagittal plane. And then the third one is the horizontal one, which is going like this. This horizontal semicircular canal is making a 30-degree angle with the horizontal plane. So it's not straight like this. It is making a 30-degree angle with the horizontal plane. Do you understand, everybody? The three planes. Now you see this one. This is the anterior semicircular canal, which is making, if this is the sagittal plane here, let me draw the sagittal plane so you can make out. I cannot. Oops. So let me draw. If you have the sagittal plane,
enable editing.
yeah, this one, right?
Enable editing. If you try now, Doctor.
The previous one.
Yeah.
So if you have the sagittal plane, so if you see this, semicircular, it is making almost of should be flat. So this is the sagittal plane. The angle which you are going to see with this one, this is not straight like this. it's making an angle of 45 degrees with the sagittal plane. So this is your sagittal plane, this is your semicircular canal going like this. So it's making an angle of 45 degrees with the sagittal plane. Let me draw it here. So you have this sagittal plane, your anterior semicircular canal is making an angle here of 41 degrees. Posterior one, if you see, this is your sagittal plane, it is going to make another angle backward like this around 56 degrees. The horizontal one is going to make an angle from the floor of 30 degrees. So it's not straight, it's like this a little bit. Now, what is the advantage of this? Your anterior semicircular canal, imagine this is y-axis. Okay? The horizontal semicircular canal is x-axis. So anterior is y-axis, horizontal is x-axis, and posterior is z-axis. So in all three, axes, these canals can detect the movement. So if you are moving towards the right horizontal canal is activated, the other one would be inhibited. So this is how the brain is going to get. You're moving towards right is basically becoming activated, left is basically becoming inhibited. Now the other thing is the anterior canal or the superior canal is going to be activated when you are moving the head like this. Your head nodding like this is basically going to activate the anterior or superior. Your head tilts this side or that side, the posterior would be activated. If you are going to move your head like this or this, your horizontal are basically activated. So head movement like this is anterior, head movement like this, like you're just tilting towards the shoulder, this is posterior, and if you're tilting this way or that way, moving the head this way or that way, horizontal is the one which is going to be activated. So this is important. You are seeing that the horizontal is basically in x-axis, anterior and superior is your y-axis, and the posterior one is your z-axis. And that's how it is going to take the stimulus in any direction your head is going to rotate. It is going to be able to detect. Now the other interesting thing is your right anterior canal, which is this one. Okay, this is parallel to your left posterior. You see, the right anterior is parallel to the left posterior, and left anterior is parallel to right posterior. They are in the same axis. If you move the axis forward, you would find that right anterior is parallel to the left posterior and left posterior is parallel to the right anterior. Okay, rotational movements of fluid inside the semicircular canals will bend the hair cells, and if the hair cells, the stereocilia, again these are going to be the similar stereocilia and one big kinocilia. If the stereocilia are going to beat towards the kinocilia, there would be action potentials generated. If they move away from the kinocilia, action potentials would be decreased or inhibited. So in one canal you would have activation, and in the other canal you would have the inhibition, and that's how the brain is going to interpret which direction your head is rotating. Okay, now what you need to understand is that in the semicircular canals you have this ampulla, and within the ampulla you have this something which is called the cristae. The cristae has these stereocilia and one kinocilia, and this is all embedded in the cupula, which is a gelatinous structure. So imagine you are moving your head towards the right. Okay, now we are talking about only the lateral semicircular canal. Okay, so I'm going to move my head towards the right. the endolymph in the my right horizontal semicircular canal would have inertia. That means they are not going to move forward, they would stay at its place. The endolymph within the semicircular canal is not moving, it has inertia because it is heavy. But your right semicircular canal is rotating because your head is rotating. So as it is going to rotate, it is going to hit the endolymph. So it seems like the your ampulla or Christie is moving towards right, and your endolymph is moving towards left because it has inertia. So it is basically kept at its place. It's not moving. It has inertia. It is basically in the canal. It's not moving, but your head is moving. So your canal is moving. So as the canal is moving, your Christie ambularis is going to hit the endolymph. So it looks like that the endolymph is moving towards left, and your Christie is moving towards right. What would happen because of that? The cupula would bend, and the cupula is going to bend. It is going to cause the stereocilia to beat towards diaphyselia, opening of the TP links, which will allow the potassium to go in. As potassium goes in, there is depolarization. Calcium comes in, causes the release of the neurotransmitter, which is the glutamate, causing the vestibular nerves to get activated. Yes.
Okay, so when I turn the right side,
Yeah.
the cooler goes to the left.
The cupula, don't talk about your cupula. Your endolymph is going to move towards the left. It looks like it is not moving. It is in stationary. It has inertia. But your right, semicircular canal is moving, turning towards right. So as it is turning towards right, and the endolymph is in inertia, your Christie is going to come and hit the endolymph. So it would seem like that the endolymph is going towards the left, and your canal is moving towards right, and that would push the cupula. And if the cupula is pushed, your stereocilia, which are basically on this side, they would beat towards gynecilia, which will cause the opening of the TP links and potassium channels, causing the calcium to get in and the release of the reaction potential. So what you are seeing there, you moving towards left, and you are seeing the endolymph moving this way, causing the cupula to move. And as it is moving, the stereocilia are moving towards gynecilia, opening the TP links and the potassium. Yes.
When I did my life,
Yeah.
the lift still is acting, there will be a gap between the still and
So in your left horizontal semicircular canal, the endolymph is going to move away from the cupula. Okay. On the right side, when you are moving your head towards the right side, the endolymph is going to push the cupula. Why? Because it is your head is turning this way, and endolymph is coming and moving towards left. On the other side, it is going away from the cupula. So it is not going to bend the cupula. Let's draw it, because that's something which you need to understand what is going to happen. So let's draw this. Let's draw the semicircular. So this is one semicircular canal. We call it the right. Okay, and then the smaller segment. This is right, and we can make this one left. Okay, and you're turning your head towards right. Okay, so what? And we make the cupula here. Let's make the cupula. Okay. So what is happening here? Your head is moving this way. Okay. The endolymph, because this canal is moving all the way to the right, endolymph is stationary. Okay, so it seems like that endolymph is pushing this way. Endolymph is stationary. It's just the canal is going to move this way. It would hit the cupula, bend the cupula to this side. Okay. At the same time, what is happening in the left canal? You're moving to that side. Endolymph, this is your cupula. Endolymph is moving this way. Because the canal is moving all the way this side. Endolymph is stationary, it's moving on the other side. So it's not going to bend the cupula at all. The only place where the cupula is going to bend is the side where you are basically moving. So this would get activated, this would get inhibited. Okay, everybody clear? Yes.
Does the activation or inhibition depend on what side the physiology works?
Yeah, so we always said stereocilia has to bend towards anesthesia for polarization. If it moves away, hyperpolarization. Yeah, so these are all fancy animation, but it doesn't tell you much. This is important because you see, head is turning this way. This semicircular canal is getting activated, and this one is getting inhibited. So your firing of the neurons in the right horizontal semicircular canal is increasing. Your firing in the left horizontal semicircular canal is decreasing, and that's how the head is basically, the brain is interpreting which side the head is rotating. Yeah.
The activation and inhibition only happens for the horizontal.
But it is in a different horizontal is in your horizontal axis. So when you are moving like this is your horizontal. When you are moving like this, your superior. When you are moving like this, your posterior.
And if you steer both of them these back together.
You have to find which one your superior and your one posterior is in the same axis. Okay, so this one would get activated, the other posterior would be inhibited. That would tell you if it is moving forward or backward. Okay, very, very technical things. So it's interesting, but if you understand it, it's fun. So this we have done, so you can understand what is happening here. Then we move to the otolith organs. We have talked about that otolith organs, the utricle and the saccule, they are responsible for static. So utricle is for horizontal head movement. You are stationary, you're moving the head this way, that way. This is utricle. You're moving the head, like you're stationary, you're in an elevator, you're going down, your head is moving down, or your head is moving up. This is vertical, and this is basically the function of saccule. Now, how it does it? It again has a sensory organ, and this sensory organ, which you see in the macula, is the macula which is present in the utricle and saccule. The macula again has hair cells. And these hair cells again have stereocilia with one large kinocilia. Above it is a gelatinous mass of which is above, which in which these stereocilia are embedded, and this gelatinous structure has otoliths. These are calcium carbonate crystals which are there making it heavier. Now, if it is going to move against with the gravity, you're moving ahead this way or that way, the whole gelatinous structure is going to move. If it bends the stereocilia towards kinocilia, again you have activation. It bends it away from kinocilia, it is inhibition. Now, hyperpolarization and inhibition, and the rest is, you can see the supporting cells, type 1 hair cells, and you have these supporting cells which are in between. This is the macula which you are going to see. Again, these fancy diagrams are useless. What is important is that you need to know where the macula is present in the saccule and where it is present in the utricle. In the saccule, it is present on the medial wall, and the hair cells are coming out like this. So they will be bent by your vertical movement of your head. Going down, going up. This is going to bend the hair cells, and this is vertical movement. Going forward, backward. This is going to move the hair cells in the saccule. The hair cells which are present in the utricle, they are present on the floor of the utricle. So they are coming like this. So when you move your head this way, that way, this way, this is going to bend the hair cells which are present in the floor of the utricle. So utricle is important in horizontal movement of your head, and saccule is important in your vertical movement of the head, based mainly used to gravity, and that's it. Now, what are the central connections of the vestibular nuclei? We know that we have, like the cochlear nerve has a spiral ganglion. Here we have a scarpa's ganglion. The scarpa's ganglion has a central process which is going to the brainstem. It has a peripheral process which is going to utricle, saccule, and the ampulla of the three semicircular gland. From there, you have the peripheral processes are going to come into Scarpa's ganglion. Scarpa's ganglion is sending the central processes as vestibular nerve, which is going to join the cochlear nerve to form the vestibulocochlear nerve, pass through the internal acoustic meatus, reach the junction of pons and medulla, where you have the vestibular nuclei. Now, there are 4 vestibular nuclei. You have the superior vestibular nucleus, you have the medial vestibular nucleus, lateral one, the inferior one. The 2 which we have talked about, which we are going to talk today, as well as are talked about quite a bit, are the medial vestibular nucleus, and the other one is the lateral vestibular nucleus. Lateral we have already talked about when we said the vestibulospinal tract. The vestibulospinal tract arises from the lateral vestibular nucleus. Medial vestibular nucleus is involved in medial longitudinal fasciculus, which connects the 3rd nerve, 4th nerve, 6th nerve with the 8th nerve. So that is important in terms of the vestibulo-ocular reflex, which we are going to talk in a bit. So the rest of them are also important, but we are not going to go and talk in detail about them. The superior and the inferior vestibular, there are 4 vestibular nuclei. This vestibular nerve, which is coming from the vestibular apparatus, is entering at the junction of pons and medulla and are supplying these 4 vestibular nuclei. The more important among them is medial and the lateral one. Now, where it is going? It is going to the brainstem. The output from the vestibular nuclei are going to the brainstem. From the medial vestibular nucleus, they are going to go to the medial longitudinal fasciculus, connecting the 3rd, 4th, and the 6th nerve, which will allow the eye movement conjugated to the head. As your head is moving, your eye is going to move. For example, if you're moving your head this way, your eye would move the other way because it is focusing on the image which it was there. So that is, and then it will be corrected. So that is done through medial longitudinal fasciculus, and that is through the connection of the medial vestibular nuclei with the MLS. It has a connection to reticular formation of the brainstem. The medial, vestibular nuclei is also connected to the reticular formation. That's why when you have vertigo, you also feel nauseating because the reticular formation is connected to the reticular formation of the medulla, which has the vomiting center. Okay. Then it also connects it to thalamus and conscious centers of the sensory cortex. It is connected, to the reticular formation. So it is important for your consciousness because through the vestibular apparatus it is going there. That's why you try to, wake somebody up, you move their head. That gives signals to your reticular formation, and it is important for your consciousness and other things. And then it is sending information to the cerebellum, which is very important organ in terms of the maintenance of balance and equilibrium. And it is also sending information to the spinal cord through the lateral vestibular nucleus, which is the vestibulospinal tract. It is facilitating to extensors and it is inhibitory to flexors. Okay, now we are coming to the fun piece, but it's also difficult to understand why this happens. So this is called vestibulo-ocular reflex. What is vestibulo-ocular reflex? Is that if you want to see that the brainstem in a unconscious patient is intact, so the first thing physician does is move the head and look at the eyes. So in a normal person where the brainstem is intact, MLF is intact, what should happen is that when you move the head this way, the eyes should move this way, the other way, because they are fixating on the target. And then they quickly correct. There is a quick nystagmus, which is called vestibular nystagmus, that quickly corrects to the side where your head is moving. And this vestibular nystagmus is a normal physiological thing. It is called saccades. If you hear the word saccades, it's a fast moving to the direction of the correction where your head is moving. This is done by something which is in your higher centers in the cortex. You have heard about frontal eye field area. So frontal eye field area is important for correction. Just remember this when we are going to talk about the pathway of the vestibulo-ocular reflex. Then we are going to talk about the correction also. The correction, the saccades, which is to the side where the eyes should, where your head is moving, is through your higher centers in the cortex. Okay. Now let's see what is happening in the vestibulo-ocular reflex. So your head is rotating to the right. Okay. So your eyes will need to rotate to the left. Okay. How this would happen? You have this left eye, right eye. We have this oculomotor nucleus in the midbrain, trochlear nucleus also in the midbrain. We have abducent nucleus in the pons. This is your right semicircular canal. So when your head is moving towards the right, it is your right horizontal semicircular canal, which needs to be excited or activated. So endolymph, head is rotating to the right, endolymph is rotating to the left, causing the excitation of your right semicircular canal. The impulses are going through the vestibular nerve, through the Scarpa's ganglion to the vestibular nucleus, the medial vestibular nucleus. This medial vestibular nucleus is going to be connected to the opposite abducent nerve nucleus. Okay, so when this is going to be excited, the right canal is excited, exciting the right medial vestibular nucleus. It's connected to the left abducent nucleus. So this gets stimulated. this left abducent nucleus is supplying the left lateral rectus muscle. Okay, so it is going to contract the left lateral rectus muscle. But at the same time, there is this MLF, the medial longitudinal fasciculus, which is going to stimulate the right MLF is going to stimulate what? The right oculomotor nucleus. So it is going to cause the contraction of the medial rectus of the right side. And then the other thing which is important is that it is also going to inhibit the right abducent nucleus, and through the MLF it is going to inhibit the left medial rectus. The result of this is going to be your eyes are going to turn towards the left. This is your slow phase. Now, this slow phase is basically controlled by the vestibular nuclei, their connection with contralateral abducent nerve nucleus, and through the MLF, which is basically connecting the sixth nerve with the third nerve nucleus. This is the reason why this is happening. Now, the connection to the side to which you are basically turning your head is happening because of your frontal eye field area. So there is a frontal eye field area, the area meets in your frontal cortex is going to send information down to the pons, and in the pons there is something called PPRF, parapontine reticular formation. PPRF. This is the center for horizontal gaze, and it is going to do completely the opposite what the MLF is doing. So it's going to inhibit the abducent on the activate the abducent on the side which you are basically turning, and it would inhibit activate the oculomotor of the opposite side, and that's going to cause the eye to move towards the side where you are basically moving. So mainly what you need to remember, correction or fast phase is through the cortex, through PPRF, through the opposite nuclei which we have talked about in terms of the vestibulo-ocular reflex. Everybody clear about this? Yes.
So this reflex happens in someone who's unconscious. We usually use this reflex to find if you get an unconscious patient. This is happening in normal people also. You suddenly turn your head, your eyes are still going to be there, and then they won't correct. But you use as a physician in an unconscious patient, if you want to see that there is no E3 to the brain stem, is there an E3 to MLF, or there is a problem in the brain stem in an unconscious patient. You can do this, just looking at the eyes, move the head, tall eye reflex, you see if it is happening, that brain stem is there.
But is there a correction or not?
Correction would be there. I will come. It will show you what is happening. Just a minute. Yes, go ahead.
The trophy nerve would be 0.62.
Yeah, so you can do this way. This is horizontal, but you can also do a vertical where trophy and oculomotor would be important.
This is again showing you the same pathway. So to and fro oscillations of the eyes, that is the function of the vestibular dysfunction. Alternating sequential movement of the eyes when the head is moving, purpose is to fixate an image on the retina while the head is moving, consists of a slow component of a track to the pursuit and a fast component in the opposite direction, which is called saccadic quick beat. Fast component is in the direction of the head movement. Slow phase of the vestibular nystagmus is due to excitatory and inhibitory inputs from the neurons of the semicircular canals to the motor neurons controlling the eye muscles. Fast component of the nystagmus is due to the excitatory and inhibitory input from the parapontine reticular formation, which is controlled by the cortex, the frontal eye field area. Now you can see it here. This is the head moving towards the right. the slow component would be towards the left, and then compensation would be towards the right. Similarly, you can see up and down also. This is trochlea and oculomotor, which are going to be involved in the lower panel. Okay, now the testing, where people use this and make it really used. So when you put up hot water in your ear, whichever the side, if you are putting in the right ear hot water, the hot water is going to cause the endolymph to rise. As endolymph is going to rise, it is going to cause the right horizontal canal. If I put the hot water in the right canal, it is going to push go to the ampulla, move the capula, and that would cause excitation. Cold water cause the endolymph to move away from the ampulla, and it is going to cause inhibition. So if I am going to put hot water, it is going to be similar that I am turning the head towards that side, away from the head towards that side. Eyes would move away initially, and then they would correct to the side. Okay? So if I am putting the hot water in the right ear, this is just like I am moving the head to the right side. When I move the head to the right side, my eyes move away, the slow phase, and then they would correct with the fast phase. This is what usually happens. Okay? So now, and in terms of cold water, it is totally different. Cold water, if I put cold water in my right ear, this means that I am moving my head to the opposite side, which is towards the left side. Okay? So my eyes are going to move towards the right side, and then they will correct to the left. Everybody agree with this concept? Because the next slide is going to be a little bit tougher. Sorry? So if I'm putting the hot water in my right ear, this means that I'm moving my head to the right side. Okay? When I'm moving my head to the right side, my eyes should move towards the left initially in a slow phase, and in a fast phase correct to the right side. So I put cold water, this means my head is moving towards the left. So my eyes will move towards right, and then they will correct towards left. Okay? Everybody agree? Okay. Now there are four scenarios. The first scenario is the normal thing. The normal thing is I have put the hot water in my right ear, so the slow phase is going to be towards the left side, and then the fast phase correction is towards the right side. Okay? This is normal. Now, if you have somebody with brainstem intact, unconscious vision, brainstem is intact, but there is an injury to your frontal eye field, for example. Okay? So, what you are going to see only the slow phase. So, when you are putting hot water, on the right side, the eyes are moving towards the left, and they would not correct back because there is only slow phase. There is a problem with the parapontine reticular fibrillation, or there is a problem with the frontal eye field area, so it is not correcting. It's an unconscious patient. You have done a calorie test. This is called calorie test, hot water and cold water, whatever you want. Now you have a person with a bilateral MLF lesion, okay, intranuclear ophthalmoplegia because of multiple sources. What is going to happen? Your connection of your vestibular nerve with the abducent, opposite abducent is intact. Okay, but MLF is gone. You put up the hot water, your opposite eye would move outward, but this eye is not going to move medially because MLF is not working. So you see, this eye, your eye on this side is not moving, the other side eye has moved out because your connection with the vestibular nuclei with the opposite abducent nucleus is working. So your eye is going to, there will be slow phase, but it's only this is going to move, this will not. Okay? Last scenario, you have a low brainstem lesion. Vestibular nuclei is gone, there is no MLF. You put up hot water or cold water, your eye will not move. So this means that you have a low brainstem. This is, for board exams, although this has no practical, but for board exams, people use these scenarios because this is a little bit difficult, high level critical thinking for you to relate first the function and then come up with. So this is a highly stuff. A lot of board exams will use these examples to test, hot water in this year, where the eye will move this way, that way, and you are basically confused what is going to happen. So unless you understand this, there's no way you can, cram it up. There are acronyms, cows, and other things which people have put up, but no way you would get confused at the end unless you understand. So it's something you understand. Once you understand, you can do it. It's easy. It's not difficult, but it becomes complicated, right? Like hot water, cold water. What is going to happen with this? So this is something challenging which you should know. Okay, last piece. Benign transitional positional vertigo. People have talked about it. Half of the vertigo lecture is going to be on this because BPD is the most common type of vertigo you are going to get. What happens in this? The otolith organs which you have when you trickle the otoconia, those otoconia somehow get dislodged and they get into one of the semicircular canals. They can enter into anterior, middle, lateral, or posterior, but most commonly they enter into posterior canal. So posterior canal is, if you remember, it is basically this or that. When your head tilts in this or that way, one of this canal is going to get activated and you start to have vertigo. So in a particular position, you start to have vertigo. So they do something called Dick's height, procedure to find out which canal is basically. Once they do the Dick's tailpipe maneuver, they will find out if this is DPP or not and which canal is it. They do an Apley's maneuver, which I would not do. Apley's maneuver is done, get the otoconia out of the posterior canal. There are other maneuvers for anterior canal and the lateral canal, but Apley's is done for the posterior canal to bring out the the otoconia from the and usually the Apley's views are hanging the head down and suddenly you put the person up and turn the head. And that's push the otoconia back into the utricle. So something like that is done. Nobody is going to ask you, this is beyond your scope, but I'm just giving you a little bit of idea that if such type is done to find out if BPD is there and which canal is affected, and then Apley's is done for the posterior canal.